Hello everyone, I'm Pratima Chowdary. I'm a hematologist at the Royal Free Hemophilia Center, and today it's a pleasure to provide an overview of the MRC GO-8 hemophilia A gene therapy study. So the GO-8 is a typical phase 1-2 study of a liver-directed AAV gene therapy for the management of severe hemophilia A. The objective was to assess the safety and efficacy of escalating doses of a novel cassette which was pseudotyped with serotype 8 capsid for treatment of severe hemophilia A. Typically, it used an adaptive trial design. Essentially, it had 4 dose cohorts. Patients could receive 6 E11 vector genomes per kilogram, 2 E12 vector genomes per kilogram, 4 E12 vector genomes per kilogram, or 6 E12 vector genomes per kilogram. The decision to dose escalate was done by an independent data monitoring committee. The primary endpoint was safety. We wanted to see the number of vector-related adverse events and also if the patients developed any inhibitors to factor VIII. And the efficacy was the secondary endpoint, and we measured the factor VIII levels both by chromogenic and one-stage assay. And we also looked at the impact of the factor VIII expression by looking at the consumption and also the annualized bleed rate. The main study lasted for about 6 months, but the idea is to follow up these patients for 15 years or potentially a shorter time frame if regulatory authorities allow us to do so. So what is unique about the cassette used in this study? The BioMarin study, if we recall, used a standard B domain deleted factor VIII transgene. In this, there is a truncated B domain which has got 18 amino acids and 6 aniline glycosylation motifs. When you see in terms of the preclinical expression, there is an increased expression compared to your B domain deleted codon optimized factor VIII. It's almost a 3-fold increased expression of factor VIII. And of course, what this means is that we can use lesser vector genomes to achieve the same amount of clinical effect. So in terms of, if we now look at who were the patients enrolled into the study, it was typically we enrolled patients who had severe hemophilia who were 18 years or older. If they had antibodies to AAV8, they were excluded from the study. If they had history of inhibitors to factor VIII that required immune tolerance, they were also excluded. One of the distinguishing features of the study was the use of prophylactic immunosuppression. We used prophylactic prednisolone, about 60 milligram, starting around 6 weeks after the vector infusion from the second cohort onwards. Tacrolimus could also be used, and typically the steroids were tapered after about 6 to 8 weeks. If the patients had an elevation of ALT or AST at any point in time, which was more than 1.5 times the baseline and were not on steroids, then you could also initiate them on steroids, which we call as the reactive immune management of the transaminitis. There were about 12 patients enrolled so far, and I think the average age is around 30, and essentially I think all kinds of mutations have been responsible for the severe hemophilia, and all of the patients received prophylactic steroids. What is noteworthy is some of the patients have had previous hepatitis C, which had been eradicated with the new antiviral therapies. Across the dose cohorts, what we can see is a dose-dependent effect. With increasing dose of the vector genomes, the peak factor VIII level increased. And then if we look at the expression in the 2E12 cohort, we can see that the expression is stable up to 4 years. And this is something that is unique to this cohort, which we do not see in the other cohorts, that beyond a 52-week period where we have seen the stability of expression, the levels continue to be the same across year 1, year 2, year 3, and up to year 4, whether measured by one-stage assay or chromogenic assay. Within the first year, you do see a higher levels, which is partly related to the rapidity of expression of factor VIII, but also with the secondary impact of steroids on the promoter. Of course, what we realized is that although increasing factor VIII expression was seen, what is of interest for us is the durability of expression. And the highest durability of expression was seen in the second cohort, where patients received 2 E12 vector genomes per kilogram. We can see there is a rise in factor VIII immediately within about a week to 2 weeks, and then again you see a further increase when the steroids are given to the patients. But typically patients' expressions tend to stabilize around 52 weeks, and then from 52 weeks onwards we've seen stable expression up to 4 years. This has not been the case when you look at the 4E12 cohort and the 6E12 cohort. Whilst the expression in the first year was higher than that was seen in the 2E12 cohort, we saw that by the end of a year and later, that the expression level was potentially lower than that in terms of the 2E12 cohort. Although once the expression stabilized, it tended to continue to be stable into up to 2 to 3 years. The 6E12 cohort had seen the most amount of immune response, with some of the patients requiring multiple courses of steroids. And again, I think we don't have expression data for all of the patients up to 4 years, but what we noted is at least the first 2 patients, one of them has done extremely well, whereas in another patient, the patient has required prophylaxis because of the immune response resulting in loss of expression of the transgene. So if we had to summarize, we can confidently say that a single infusion of this novel V3 variant resulted in a stable expression beyond the period of transaminitis and immunosuppression. So essentially we are saying we can see stability of expression around 52 weeks, and beyond this period, particularly in the 2E12 cohort, we've seen stable expression up to 4 years. With the higher dose cohorts, that is the 4E12 and the 6E12 vector genomes, whilst the peak levels were much more higher, what we've not seen is the stability of expression beyond the 52-week period. But more importantly, the immune response was so vigorous that the loss of expression resulted in the levels that were lower than what were seen in the 2E12 vector genome per kilogram across the entire cohort. Importantly, 9 of the 12 participants have been able to discontinue factor VIII prophylaxis, and the idea is now to increase the recruitment to the 2E12 vector genome cohort to see the consistency of effect across a larger group of patients. Thank you for your attention.